Welcome to the lecture series on antigens. In this video, we'll see definition, attributes of antigenicity, types of antigens, and factors determining immunogenicity. Let us start with the definition. The antigens are the substances which, when introduced parenterally into the body, stimulates the production of an antibody with which it reacts specifically and in an observable manner. So, can you call any substance as an antigen then? No. To behave a substance as an antigen, it should show some properties, and these properties are called as attributes of antigenicity. Number one is immunogenicity. It is the ability to induce immune response. It may be antibody production, that is humoral immune response, or production of sensitized T lymphocytes, that is cell mediated immune response. Number two is immunological specificity. The antigen reacts with a specific antibody or sensitized T lymphocytes. Based on these two attributes, antigens are classified into two types as complete antigens and incomplete antigens or haptens. Complete antigens are able to induce antibody production and produce specific and observable reaction with the formed antibodies. Therefore, complete antigens are immunogens. All immunogens are antigens, but all antigens are not immunogens. Incomplete antigens or haptens. They are incapable of inducing antibody formation, but can react with it specifically. The haptens or incomplete antigens can be converted into complete antigens by adsorbing on the surface of carrier molecules. There are two types of haptens, as simple haptens, which are characterized by simple chemical substances. They are monovalent. They can react with the antibody, or they are unable to precipitate the reaction. Second is complex haptens. They are large molecules. They are polyvalent. They are able to react with specific antibody, and they are able to precipitate the reaction. Examples of haptens are penicillin, sulfonamide, aspirin, neomycin skin ointment, some cosmetic, and tranquilizers. On the surface of antigen, the smallest area which determining the immunogenicity is called as epitopes. In the diagrammatic presentation, the green colored areas shown, they are epitopes or antigenic determinants. So it is the smallest unit of antigenicity. They are immunologically active regions on a complex antigen. And antigens are multivalent in nature. It is the smallest area on the antigen, usually consisting of 4 to 5 amino acids or monosaccharide units. The properties of epitopes are size ranging from 25 to 35 angstrom units, molecular weight ranging from 400 to 1000 Daltons. The determinant groups present depends on the type of antigen. For example, in case of protein antigens, the determinant groups may be penta or hexapeptide, that is containing 5 to 6 amino acids. If the antigen is polysaccharide, it may be hexasaccharide in nature. There are some factors to determine the immunogenicity. These factors are: number one, foreignness. Our immune system has ability to recognize between self and non-self. So non-self substances, they are called as foreign. Antigen should be foreign to the host. Generally, the greater the phylogenetic distance between the two species, the greater the structural disparity between them. For example, bovine serum albumin injected into cow is not immunogenic, but bovine serum albumin injected into rabbit is found to be immunogenic. There are some exceptions to this. For example, collagen and cytochrome C, they are not immunogenic because they are highly conserved throughout the evolution, whereas the self components such as corneal tissue and sperms, they are found to be immunogenic. Second factor is molecular size. The active immunogens have molecular mass of 1 lakh Daltons. Substances with molecular mass less than 5000 to 10,000 Daltons are poor immunogens. The substances having low molecular weight can become antigenic by adsorbing on carrier particles such as bentonite. Third factor is chemical nature. Proteins are found to be highly antigenic, but not all proteins. For example, gelatin, histones, and protamines, they are not immunogenic. Chemically complex molecules are more antigenic. Substances of diverse chemical and structural properties They are found to be most powerful antigens. Presence of aromatic amino acids increases antigenicity. Fourth factor is susceptibility to antigen processing and presentation. The substances which are phagocytized in the body are immunogenic. Large insoluble macromolecules are generally more immunogenic. Large molecules are more easily phagocytized and processed. The polymers of D amino acids are found to be poor immunogenic because D amino acids are not metabolized in our body. The fifth factor is route of administration. The route of administration strongly influences which immune organs and cell populations will be involved in the response. Antigens entering in the body via parenteral routes are more immunogenic as compared to oral route. Post your suggestions or questions in the comment section below. Subscribe my channel and like my video. Thank you for watching my video.